this regulation in the body that is making the progression of other diseases, also the survival of that. This is one example of a software system that is available. You can look at that how you can use that model and then develop the system for identification of biomarker and the progression of the disease identification. So in this way, we have developed several software systems as well as this is one software system that is also published, but it is not available till now because it is going to patent, so that is not available. So another one is that is mentioned and also Cytochrome also another software to understand your functionality of the genetic marker for the identification of the diagnostic biomarker. And so that is the take-home messages. I'm not taking too long time, so maybe almost, I'm not sure, maybe almost 15 to 20 minutes past. So the take-home message is for diagnostic biomarker identification, we need to take care about it, not only consider the single genetic marker. We need to consider multiple biomarkers. That means different, we can say different database. Also, we need to think about the confounding, maybe one biomarker set could be interact with different diseases. So we need to think about that. So we need to identify the unique set of biomarkers. So that is the reason we are developing the system considering the different type of biomarker, and the algorithm is of course the advanced deep learning algorithm or RDO identification. And finally, we developed a couple of central systems, and still we are developing that for the diagnostic and prognostic purposes for human. Also, we are using these type of biomarker for developing different sequences like the biosensor or also different kind of not only the sensor, also different kind of software for the diagnostic and prognostic. Thank you so much. And overview of the project that I explained, actually many universities and institutes are involved, and a lot of collaboration, including in Bangladesh. So that is also in some universities from UK, USA, and Australia, also many universities in Bangladesh. Thank you.
Thank you, sir, for your wonderful speech.
Okay, thank you, Dr. Moni, for your presentation. It is informative, and also there are many things to do, I think, in this domain. So now I would like to ask our audience if you have any questions to our Dr. Moni. Okay, please.
Yeah.
Dr. Mohammad Ali Moni, a very exciting presentation. I am Tufajul Islam from Gazipur. The model you developed, is it possible to use in agriculture also to trace the animal as well as the plant diseases and their condition?
Thank you so much. Actually, it depends on the data. So the main concern is the availability of the data. So the system is, of course, when we consider the animal, for example, I can see the more related to animal, human body and next to animal. Then we can go for plant and crops. So the animal, in any case of animal, we need to think about that. Also, for example, diagnosis, I don't know the diseases of the maybe cow or many diseases are available. But we do not take care as human because maybe if one cow dies, then it will be loss of money. Okay. So in this case, it is applicable. Of course, applicable. But my concern is for the diagnostic purposes. My main focus is, okay, you need to, for example, I can give ovarian cancer. There are 60 biomarkers for the testicle. But all 60 biomarkers, they are take dropping. They all are not used for diagnosis. That is my concern, my point. So how can you identify these biomarkers? Same in case of animal, in case of plant, it is same. I can say same. All the many biomarkers you can identify. You can publish it in your lab. You can show that this is the biomarker, but not diagnostic biomarker. Because maybe for maybe rice, there is a maybe some, I forget the name of the disease of the rice. Maybe one disease that is maybe related to many biomarkers, maybe related to that disease. Maybe for when we will say some diagnostic purposes, maybe we cannot use all the biomarkers. for one when we consider one data. So we need to identify the small set but unique set that are only influencing data, that are only other reason related to data. That is one point. Second point is, of course, in case of maybe band or cross, it is not suitable. We need to consider multiple types of data because the epigenomics, maybe also you know that the epigenomics, maybe that if we identify the epigenomic biomarker, that is good. That can be changeable and modifiable and useful. But epigenomic marker can cover its on side, on type of like on group of problems. But maybe we need also the transcription or genetic marker that can be useful for maybe another group of problem identification or diagnosis purposes. So if you consider combinedly, then it is useful. Maybe one epigenetic marker, one mutation or two mutation, on the other hand, one transcription. Combining, if we develop device, that will develop perfect. That is my point. So that what this do is we need to identify small set of biomarker that is unique. Second is we need to consider as many types of data. Then we can consider all the types of data that should be combined in a small set of diagnostic parameters. So that is very suitable for the diagnostic and prognostic.
Thank you.
Of course, the final point is I am coming to you that of course it is useful. The process, the method that is developed for the human, it is also applicable for the animal and plant.
Okay, thank you, Professor Moni. So any other question from the audience? Okay, please.